Good afternoon, everyone. So welcome to this EURASIP webinar. My name is Geert Leus, and it's a pleasure for me to introduce Dorina Thanou from EPFL, where she leads strategic initiatives on AI for health. Dorina also received her master's and PhD from EPFL. Her focus is mainly on graph-based signal processing and machine learning, with an emphasis on interpretability and robustness, and as application focuses on biomedicine, as we will see also today. Also, one of our aims is to bring in biological and clinical domain knowledge into AI structures. So we will also see that today in her presentation. She received several awards for her work. She's also a member of the EURASIP Technical Area Committee on Biomedical Image and Signal Analytics, and a steering committee member of the Data Science Initiative of the IEEE Signal Processing Society. So it's really a pleasure to have her here today. I know Dorina for many years. We actually organized also a webinar together on graph signal processing. It has been a while since we did that. But since we organize it together, we never have the chance to introduce each other for a talk. So it's a pleasure that I could do that today for this EURASIP webinar. So okay, so it's time to get started. So for the questions, try to either reserve them for after the talk or put them in the chat, so we can handle them after the talk, so we can have the recording clean without interruptions. So go ahead, Dorina.
Thank you, Gert. Thank you for the nice introduction, and thanks to you and Nelly for the invitation. It's a pleasure to present today a bit of the work that we are doing with a large team of collaborators, students, etc., that is broadly in the domain of AI and medicine. There will be a strong emphasis on graphs in this presentation, and the idea is mainly to show you and hopefully convince you that different use cases, different instances of medical problems can fuel the design of novel AI algorithms. So it will be more on the depth, on the breadth side than on the depth side, but I'm very happy to take questions and go into more details in specific part of the talk that might be more interesting for the audience. So let me first start by saying that nowadays, of course, the great technological advancements are transforming medicine. And that's clearly because nowadays, for each patient, we have an atlas of data coming from different technologies. Each of these technologies is capturing different aspects of the disease. For example, pathology, you can go at a cell level, check the structure of the cell. Radiology, you can get your CT scans, PET CT scans, full body scans. At a single cell level, you can go and understand the genome, the genomics. Multi-omics technologies, you can also have information at different scales, even going at a 3D. So all these technologies are now much more developed, which helps us believe that with this wealth of information, if we can exploit it correctly, we'll be able to understand better different disease mechanisms, we'll be able to produce tools, assisting tools for clinicians or medical scientists, biologists, in order to empower them in their decision-making process, but also at the same time lead to novel discovery, biomarker discovery, etc. So we have all this data, and of course we have the great advancements in terms of AI nowadays, but is it an easy problem? And of course, the answer is no, and I think that's one of the most interesting domains where AI can have a significant impact, mainly because of the particularities of this domain. If we think of health globally, the problem is quite challenging because health is fundamentally multi-scale. You have all these different data modalities that we mentioned before, each of them capturing different aspects of the disease. So you start at the cell level, you go into the environment. If you focus on tumor, for example, interaction between different cells, the tumor environment, but then you can go also to realization of the phenotype in terms of the full body, lesions appearing in the body, etc. So you have different interconnections, different interactions between genes, each of them changing every time, changing the identity of the cell, the phenotype of the cell, etc. And all this happening across time. So we have a very challenging ecosystem where at the same time we have multimodal data, multi-scale dependencies, and we have a time component that is quite important. And said that, we are in a regime where, of course, we are not talking about classifying cats and dogs. We are talking about small clinical cohorts, in particular in diseases that are rare, where fortunately we don't have a lot of data samples because people are not sick from this disease. So it's quite a very challenging domain. It's quite a challenging ecosystem where definitely AI can bring a significant benefit for society in general. If we think historically of how AI has been designed and generated across the last few years, we have started by designing models by taking into account the right inductive biases. For example, if we think of perceptron, of how it started, we wanted to approximate regular functions. Convolutional neural networks, people started with the observation that in images would like to take into account the notion of translation invariance. So whenever one object appears in one part of the image, we should be able to recognize that that object that appears in a different part of the image correspond to the same thing. So this notion of translation invariance led to convolutional neural networks, the definition of convolution, etc. RNN, there is a strong bias into the time components. And then more recently, graph neural networks, we have the notion of permutation invariance there across different nodes of the graph. So all these architectures, these are great development in terms of AI, and they excel in different use cases, but in particular in use cases where these inductive biases are true. Now, if we move into biology and health applications, some of these architectures can definitely be used and should be used, but at the same time, there are additional complexities that are coming from the fact that, as we said, biology is multi-scale, biology is changing quite a lot over time. You have image technologies that are not RGB. You can have more than 40 channels, for example, in order to be able to capture the expression of different proteins in a cell. So how can you exactly capture these particularities in the health space, and how can you translate what we have as methodologies nowadays in terms of machine learning for medical applications? And that's, of course, that's not an easy task, and that's an effort that many people, and I'm sure also people from the audience here, are working towards how to enrich current AI models with inductive biases coming from the medical structure. So the idea would be to try to build AI algorithms that would move from human design models to models that are data driven, but also specialized with domain knowledges or inductive biases coming from the application domain. And that, of course, what we want to achieve is to have this set of tools that on the one hand would provide assisting tool support to clinicians in their decision-making process, diagnostic tools, prognostic tools, etc., but also lead to novel discovery in biology. Identify novel biomarkers, identify different combinations of cell types, and interact with each other. So it sounds quite theoretical, but just to make it a bit more concrete, and in particular the type of questions that we are trying to address, we will focus in one specific aspect of inductive biases, that is about graphs and networks. So networks can be considered as a natural and quite powerful inductive bias for quite a lot of biomedical applications. If we think, for example, of a brain, there is already a structural connectivity coming from how neurons are connected to each other. In terms of spatial biology, we know that cells interact with each other. And in particular, where they are organized in space, it's quite important. So there is quite a strong notion of, or indicator that networks could be useful in order to approximate the spatial communication between cells. Protein-protein interaction, of course, these are quite well-known networks. In terms of molecules, we have different atoms interacting with each other, gene regulatory networks, but also if we go into geometries, if you focus, for example, into the heart and the coronary artery, the geometry, it's a structure. And this structure can be approximated by meshes, can be approximated by graphs, etc. And broadly speaking, more broadly speaking, you can have also complex dependencies between different diseases, between, as I mentioned before, between different atoms or items at different scales, comorbidities, different genes that are interacted to each other, etc. So in all these network type of applications, graphs are a nice mathematical tool to model this sort of data. And basically, to make it even more concrete, the two big cases that we would focus today, and that's the research of the main focus of our research, are the two largest diseases, that is cardiovascular disease and cancer disease. I guess I don't need to motivate them too much. Unfortunately, most probably most of us will have or had someone in the closest circle suffering from one of these two diseases, and it's a disease that these two together, they account for 50% of all global health. The numbers are quite huge in terms of how many people suffer from either cardiovascular diseases or cancer nowadays, and also in terms of how many people are dying per year globally. So of course, if we can have some impact in terms of these two disease cases, or even one of them, that could be a very nice contribution of AI to the society. So what we will focus about, so basically we will present, I will try to present some methodological development in the space of graph machine learning, and in particular how these developments are associated with different instances of health problems, if you want. So I will focus in three different use cases. The first use case is at the tissue level, and we will try to see how methods from graph machine learning can be used in order to understand better the tumor microenvironment. Then we will move into cardiology, where aspects such as physics-informed knowledge, digital twins, etc., can be used to predict future cardiovascular events. And finally, I will finish with moving into temporal aspects, and in particular, how we can model tumor dynamics over time, and how can we use this modeling for better treatment of patients. So this would be the three parts that the talk will be divided. So let's start with understanding the tissue microenvironment. I will give a bit of background because I'm assuming that not all of you are working in health applications. So understanding the tissue complexity, it's quite challenging, it's quite important. Because there we have interactions between different cell types. And what we mean by cell types, of course you have tumor cells, but then you have also immune cells, different types of immune cells, macrophages. You have stroma, that is the environment surrounding the tissue, etc., etc. So you have, I will not go into the details because that's not the purpose of the talk. You have a very complex ecosystem of different entities, different cell types. Each of them has a specific function. How they interact with each other will define how the patient will respond to specific therapies. If a drug is appropriate for a specific disease, and how the patient will evolve in terms of metastasis, if the tumor will be metastatic or not. So capturing these interactions between the cells in the tumor microenvironment is quite important, but at the same time, it's quite complex. There are quite a lot of works nowadays, and in particular, if we focus into foundation models, etc., people usually are building large foundation models, mainly from imaging. So some of the main modalities in which we capture the tumor microenvironment, first of all, is H&E. That's the cheapest modality in which a pathologist is using in order to identify cancer cells. But then you have other sort of modalities that are coming from multiplex immunofluorescence images, are the images on the left that you see here, the colorful ones, where each color will identify a specific type of cell. So abstracting a bit from the cell, you can think of these colors as clusters of nodes, basically, which represent a different sort of information. So there have been a lot of work in terms of modeling this data using vision-based approaches that of course are a bit domain agnostic in comparison to the structure, to the biological structure of this data. So our hypothesis is that if we follow a cell and the spatial aware modeling, so if we focus on the core biological entities that are cells, we would be able to guide the learning into more biologically meaningful features. And to be a bit more concrete, we would like to move from image-based representations, where you basically take a large image. These are usually whole slide images that are quite big in terms of gigabytes and or in terms of how many millions of cells you see there. So usually what we do in vision representations, we take these patches and then we analyze these patches with vision-based models. Now I would like to move one step further. Would like to get rid of this artificial patch construction, but by building constructions that are guided by the structure of the tissue. And the structure of the tissue meaning the cells that are the key components of this tissue. So basically we move from the image into identifying, segmenting the cells that can be nowadays very easily done with existing tools. And each of these cells will be represented by a node on the graph. And then we design edges that are biological relations into the cells. And basically what we're interested in is mainly spatial relations. So connections between cells that are close to each other in order to be able to understand, for example, if a tumor that is full of tumor cells is surrounded by immune cells, if that would have a better prognosis or not for a disease. So all these graphs, of course, can be used for those of you who are familiar for tools such as graph neural networks, graph signal processing sort of tools in order to be able to model the spatial organization of tools of these cell-based representations. So there are a lot of people working in that domain. There has been quite a lot of work, but of course it comes also with the limitations of what we know coming from the design of graph neural networks, for example. They are good in capturing pairwise relationship between nodes, but of course, if we want to go into larger scale dependencies, things can become quite tricky. So one specific angle that we are trying to investigate is how to go beyond spatial neighborhood and how to be able to capture also the topology of the data. So if we have spatial structures in the data that could be clusters of cells or different circles around a specific area, we should be able to identify that. And how we are doing that is by using tools from persistent homology. So topological data analysis in general and persistent homology in particular are tools that allows us to identify these shapes in the data. So we start in particular in the example that I mentioned before in terms of cells, we start from at a cell level, or it can be a cluster of cell, tumor buds, etc. And then we try to model through simplicial complexes interactions at different scales. So basically, these cells would be considered as point clouds. And then we try to build filtrations of these simplicial complexes by using different filtration functions such as Victorious strips, where you try to identify in increasing radius around a point or a cell, and to see how specific shapes appear or disappear over time. So if we have cycles appearing, if we have triangles appearing, etc. So we have all these different simplexes appearing over time. And of course, we can define what we call barcodes that will reveal where these simplexes appear and disappear across different scales. So persistent homology can be a way to capture this information at different scales, and in particular to capture it with respect to the different pairs of cell types. So that's a joint work that is in collaboration with Bernadette Solz. She's an expert in topology. And basically what we're trying to do there is to build a graph learning algorithms guided from topology for spatial biology. Basically, the intuition, the idea is that we'd like to combine local multi-species witness features with GNN architectures. So the way that we do it is that we build persistent homology features. The way that we build it is through some witnesses in order to be able to take into account persistent diagrams per each cell type. And then in the end, we compute distances between these persistent diagrams in order to be able to see what the distribution and of each cell type in neighborhoods of different scales. Once we do that, the details, we can go into more details later if there is a question on that. But once we compute these topological features, basically these features can be considered as as features on the node of a graph, where a graph is always spatial. It can be a Delanoid triangulation, where, for example, you have nodes as cells and their connection, spatial connection as nodes, as edge on the graph. So we can think of these features and as some sort of positional encoding coming from notions from persistent homology, in order to be able to capture the distribution of the cells in terms of different scales and in terms of the tumor microenvironment. So just to give you an idea of how this sort of methods, technologies perform, we have tested it on a synthetic data set that is an agent-based model. That is a model. It's always easier to work with synthetic data sets, in particular in spatial biology, because you remove all the batch effect that is connected to different ways that different tissues are stained, etc. So these agent-based models, these are mathematical models that are designed with the mindset of how we know that the microenvironment functions. So we have different cell types, macrophages, blood vessels, necrotic cells, tumor cells, etc. And then we have different states in which the tumor microenvironment can appear. So such states could be an equilibrium, for example. The equilibrium is a relatively good trade-off between the tumor cells and the macrophages that are fighting the tumor cell. We have the invasion that is a status that is not necessarily what we want, meaning that you have a lot of cancer cells invading in the body, and then you have the elimination, meaning that the macrophages have eliminated the tumor cells, which is a good prognosis in general. So we have these three artificial but realistic scenarios that help us understand is indeed these topology features integrated with the spatial aspects coming from the GNN can be promising. And indeed, that's the case. So I will not go so much in every comparison, but we have been comparing with different methods, other coming from simple ML methods where we would aggregate simple statistics of the features, etc. Methods that are coming more from the topology aspect without any GNN involved, and also methods that are purely GNN related with no topology there. So we can see that we can indeed achieve a much better performance by combining topological features with features with a GNN architecture that is enhanced with this positional multi-scale encoding scheme. And then in terms of interpretability, we can see that we can indeed get some more interpretable patterns that are associated with each of these stages of the disease. I will not go too much into the details of that, but after discussions with clinicians and biologists, we can see that indeed the method is focusing into patterns or clusters that are indeed meaningful for each of these categories, classes of applications. In collaboration with the University Hospital of Bern, we are working also in colorectal cancer dataset, where basically the idea is to try to predict from the cell, the cell organization and the cell type, if a person is going to relapse or not in the future, which it's an insight for a surgery. And basically, again, the conclusion is more or less the same, that you get quite good behavior already with topological features, but then when you introduce them into a GNN architecture, you can get an even better performance. So that was the first part that was more at understanding at the cell level, at the tissue level, the organization of different cells. And basically the priors there that we discussed into designing the algorithms are coming from the fact that spatial aspects are important. That's why we model them with graphs. But also at the same time, we have the information that the shape of different organizations of cell type is important. So that's where topology comes, and what's there, the interplay and the interaction between tools coming from graph machine learning and tools coming from topological data analysis could be quite impactful. So we will move in a slightly different application where now the priors are completely different. The use case is in cardiology, and the prior basically that we know from the application domain is related to hemodynamics, physical equations, etc. To make it more concrete, so of course, cardiovascular diseases, as we said, are the leading cause of mortality nowadays. The basic, very basic modality or image technology that people would get when they go with some symptoms or a heart attack in clinic is coronary angiography. That's how it would look like. So it's this, it will capture it's this imaging modality that will capture the structure of the arteries and will tell you if there is a narrowing, the red box there, of the artery or not. Because of course the narrowing or stenosis, as we call it in the medical context, it's an indicator that something is wrong, and then they have to intervene and they have to put a stent, etc., etc. So it's an invasive modality, which means that in this case, you cannot have a lot of data sets because you don't direct patients for an ICA, for an angiography, when there is no strong reason. So our vision is to be able to define AI-based tools for preventing from cardiology, meaning that we would like to predict if a person is going to have a heart attack in the next 2 or 5 years. And how do we do that? Is by combining data, of course, but together with medical knowledge, but also knowledge coming from physics, physical knowledge, what we know from hemodynamics, etc. So to be a bit more precise, so as I said, the narrowing of the stenosis is the first thing that the cardiologists would look with these modalities. And basically one way to capture that geometry is by being able to identify that shapes and that shape and model that shape with a graph. So what we have done is to take these images, these are 2D images, annotate them with the help of clinicians and be able to identify these structures that are capturing the geometry through the graph. Once you have this geometry, there are different parameters, of course, that you need to take into account, how many data points, how dense the graph could be, etc., etc. I will not go too much into the details of that, but you have a graph capturing the geometry of the structures. Once you have that, then you can fit a graph machine learning model that would basically be enriched also with clinical features that you might have in practice, such as the type of lesion, the pressure drop, that is another measure that it's a clinical index that people use in practice for being able to identify, to treat patients. And you use this graph representation enriched with these clinical features in order to be able to predict if a lesion is going to lead to a heart attack or not in the future. And you can extend that, we have extended that also to a patient level prediction, meaning that we don't take into account only a single lesion, although in practice every patient is treated for a single lesion, but we move also into patient level predictions, meaning that we combine different lesions together. So we have built a mask attention model in which you are able to adapt to a different number of lesions per patient and have a score that will predict if a patient is going to have an event in the future or not. So, and the results are quite promising. Again, I will, for the sake of time, I will ask you to trust me that by using this graph representation scheme, you can have a much better performance, both in terms of lesion, but also patient level prediction in comparison to purely image-based methods such as RedNet, but also classical methods such as the diameter stenosis that is measuring how big the diameter is, or the FFR that is a measure of the flow of the blood flow, that are the two indexes that clinicians use in practice. So by modeling the geometry of this narrowing of this artery tree through a graph, you can get a quite better performance in terms of future event prediction. And we went one step further, saying that yes, you can approximate the geometry from 2D, but basically you can even go into 3D. So the idea is that we take these 2D images, we reconstruct their 3D representation. This is work done by some of our colleagues here at the Center for Imaging at EPFL, where they reconstruct 3D models of the arteries from 2D images. And the reason why that is important is that through this 3D reconstruction, of course, you have a more detailed reconstruction, the spatial aspects are more visible, but also you can run a lot of simulations in terms of physical simulations, hemodynamics, etc., which leads to a better characterization of the stenosis or the patient in general. So we have what we call these 3D digital twins, in which we want to use for improving even further the prediction results that we mentioned before. The good thing with having these 3D digital twins is the fact that then you can run, as I said before, computational fluid dynamics, which of course are quite expensive. So it takes a lot of time to run this hemodynamics. It's something that some of our colleagues here at PFL are doing. And there is also a trade-off between how much this hemodynamics can help you on the types that it takes and therefore that it takes to generate this hemodynamics. The approach that we are following is slightly different. So basically, we are not computing hemodynamics, we are basically approximating them by using physical priors. We know from physics, and we know that there is quite a lot of literature in terms of how we can model fluid dynamics in the blood. Navier-Stokes equations have been used quite a lot in 3D, but also their approximation in terms of 1D. Also, the pressure blood can be modeled for healthy segments, but also for unhealthy lesion-based segments using specific physical equations. I don't have time to go into the details, but these are things that are well known in the specific literature. So there are physical models capturing these two components. So what we thought to do with that is that why don't we use these physical models, these priors that we know from the hemodynamics, in order to be able to learn features that are not an exact approximation for the hemodynamics, but are kind of aligned with what we get as are aligned with the physical constraints that we want to impose. And why do we want to do that? We want to do that because that could be an excellent way for pre-training the models, for introducing this prior in the model that we know that is valid in such a way that whenever we can do that for a lot of data where we don't necessarily have clinical outcome from patients, because these clinical outcomes could be quite expensive to obtain. And we use that to pre-train these models that would eventually be fine-tuned in terms of the final event prediction in smaller clinical cohorts where we have exactly knowledge if a person survived or not, or had a heart attack or not in the future. And to be a bit more concrete, the framework that we propose, it relies, as I said, into an image to digital twin conversions. From 2D images, we go into this real digital twin. We use these digital twins in order to augment this data set with a procedure that is inspired from this specific domain. So basically, we take randomly two digital twins, we extract the center line, and then we augment it with the radius, because we know that the radius is quite important in these arteries, to artificially generate digital twins with different morphologies. And that leads us to a large data set of digital twins in which we can do this physics-informed pre-training. Once we have this synthetic data set, we construct a graph to model the 3D representation of the digital twins. We build the machine learning framework that is built on graph neural network architectures, which we pre-train with these physical losses that I mentioned before. Once we have this pre-trained model, we use that with real clinical digital twins, and we use it for smaller cohorts, for fine-tuning, for specific downstream task that is about predicting future events. Again, some results to give you an indicator of how it works. So we have compared with methods that are similar to what I described before, meaning that you define the graph based on the 2D representation, not necessarily the 3D representation. And we can see from the blue curve on the right in terms of ROC curve, but also the precision recall. So we can see a significant gain by introducing this notion of digital twins and also enhancing them with physics-informed learning. In order to identify a bit which component is more important, we have done a very extensive study in terms of which components are important. If you focus on the subfigure D, for example, the ablation study there is with respect to the physics pre-training. So we can see that in pre-training with physics helps significantly in that case. Also, an ablation with respect to the backbone, if that backbone is 3D or is just a center line detection. And we can see that all these components together, they boost the performance of the algorithm. I don't have time to go into all these details, but I think the key message there that I want to convey is that by doing this digital twin representation, you get at the same first a more personalized representation of the patient, in which you can approximate hemodynamics through physics-informed, self-supervised physics-informed learning and get better predictions through some fine-tuning with smaller cohorts that we have in practice. Again, the same message by trying to see if the virtual data, this data augmentation with virtual teams helps. And again, in terms of scores, the higher the better, of course. We have accuracy, F1 score, AUC, the ROC curve, etc. So we can see that with all these different matrices, the green curve that is our method enriched with virtual data gets much better performance. The orange one is the same method, but with no data augmentation, with no synthetic data set. And we see that indeed we can have a boost in terms of performance in that domain. And the backbone down is just on real data. So no pre-training, but training on real data, which is much lower in terms of performance. And then the nice thing is that by introducing these physics-informed constraints, basically you end up with schemes that can give you some physiological interpretability. We are able to identify that the key features can indicate are equivalent, are aligned with what we know from biology and knowledge of hemodynamics, that basically the pressure will drop whenever there is a lesion or a stenosis, as we call it, as you will see in the boxes in red there. So there is this discontinuity in terms of pressure drop, which is reflected also on the features that we learn from the data. And also this drop is much higher whenever there is a lesion, and it's lower whenever there is no lesion, basically. And that's intuitive with what clinicians would expect in practice. So for the next 5 minutes or so, I will move in the last part of this talk that is moving into a completely different scale that is about full body scale. With a cardiology, that was an example of how we can inject physics as prior knowledge. Here, it's a completely different setup because we're talking about metastatic lesions. So PET-CT images, scans in general, are the modality that whenever someone is diagnosed in tumor and we want to follow up in terms of how the tumor evolves, someone will go and will do a PET-CT scan. So they help these modalities help us to identify how the lesion will spread on the body. And depending on that, of course, these lesions are quite complex, complicated. The mechanism behind the spreading is not known. So basically what we're trying to do is to understand the mechanism behind the evolution of the tumor subject to a treatment. So whenever a patient is given a treatment, we know that this patient will respond or not, depending on the characteristics of the tumor. And then we might have lesions that will split, will grow, lesions that will appear, lesions that will disappear. So it's a very complex and at the same time exciting environment for AI in order to be able to develop this setup, because you have all these irregularities that you don't have necessary in other use cases. So we have metastatic lesions, so lesions that appear in the full body. Over time, they change over time because there is always a treatment or there is the evolution of the tumor that is natural. And we know that there are different stages of this evolution. So you might have lesions that in some stages might seem to disappear but then appear again. You might have a disease that might be progressing. You might have a disease that might be pseudo progressing, meaning that it's a fake progression that in the end the patient will survive. So these are complicated hidden stages in which we need to take into account in order to be able to have a better assessment of the patient and be able to identify if a patient is going to respond to future therapies or not. So understanding these regimes is quite important because it allows us to understand how the system will evolve over time. So this systemic prior is exactly what we are trying to inject in our design of the models. So we try to model lesions as nodes where the characteristics of these nodes will be features coming from radiomics, for example, that are characteristics of the imaging of the tumor volume, etc., at a specific lesion. I will not go into details because it gets a bit technical in terms of the medical knowledge that is needed there. So we have these nodes that are lesions, and then we have a graph of lesions that can be considered as a system state at a specific point in time. So the goal is to be able to capture the evolution of the system by modeling the disease evolution as switching between different latent regimes. So if we switch back into graphs now and forget about the regimes, basically we have different graphs, a sequence of graphs, and we would like to identify when the sequence is changing, the state of this regime is changing, and also be able to predict future features on the nodes of the graph. How do we do that? So we build a model that relies on graph learning with variational encoders, but by unifying also state space models. And to be a bit more precise, basically we have this input data that is a set of graphs. We built a graph LSTM sort of method that will compute these temporal embeddings. And then we learn a prior that will capture the evolution of the states in the latent space. This prior would be there to modulate a decoder that would be a mixture of experts that would be able to predict the future states or the future features of the node. So we have this latent space that basically consists of interpretable latent space that would modulate the output, meaning that would be able to predict the next time point in terms of features of the node of the graph. And that's a quite nice framework because it works at inference. At training, we try to approximate this prior. At inference, we try to approximate. It can work as a state space model when we have a state transition and an emission transition, in the terminology that were used in the state space modeling. And this state in our application domain will mimic the patient states that we mentioned before. Very quickly into some results because we are running out of time. So we see that we have created an artificial data set that mimics a bit this lesion performance, but with different time steps. So with each time step, we have different lesions changing over time, some appearing, disappearing, etc. And we have also a clinical cohort, the Leticia cohort that I'm mentioning here. We have compared them with classical graph state space models, but also models that are purely GNN based, etc. And we can see that we are able to achieve better tracking and infer the patient trajectories through the states. And I will just try to highlight the fact that in this examples, if you focus, for example, at time TP5, which is time step 5, there is a big jump there because there is a transition in terms of the state. And we can see that the red color in terms of the trajectories are wrong predictions of the state trajectory, and the green ones are correct predictions. So we see that whenever there is a jump, the system takes some time, but eventually converges to an approximation of the correct state, which is quite promising and quite impactful. So, to summarize, I hope I managed to convince you that in medicine, in all these health applications, it's quite important to take into account the correct inductive biases, the correct prior knowledge that we might have from different application domains. These are application domains that are quite complicated. You always have to work in very low data regimes, and you want robust methods that can work in these very different data regimes. So, of course, graphs are one way of approximating some specific priors. They are not the solution to everything, and by no means the message of this talk is that graphs can be there to approximate any biological structure. Graphs are useful when the structure is biologically grounded. And I hope I managed to convince you that with these use cases, um, graphs can be a nice tool to develop AI algorithms that can be enriched with knowledge coming from a clinical domain or the specific medical domain. I think the domain of AI for medicine, AI for health in general, it's a quite interesting and a very interesting playground for developing the next generation of AI models. But at the same time, it's a playground that can help us advance in terms of understanding diseases, identifying biomarkers, and moving further in terms of therapies and saving people's life. Of course, that requires a lot of collaboration, that requires a lot of back and forth with clinicians in terms of what is the prior knowledge that we would like to make sure that our system has embedded in the architecture, but also to convince clinicians in order to design data sets or clinical cohorts or case studies that basically are appropriate for AI. And just I would like to finish that we are working in that direction in trying to scale these models and enrich them also with other sort of modalities. So far, what I present is our small use cases of specific clinical questions, but basically we're trying to move into translating these algorithms into real clinical impact with larger collaborations. For example, in the domain of precision oncology, we have a national AI initiative that was launched a couple of weeks ago. That is a 4 years program on precision oncology, and the idea is to be able to introduce AI along the patient journey. We are very excited about that because it's a really a national effort. And for this sort of research, for this sort of tools, in order to be able to get a pragmatic outcome, we need this sort of big efforts. And then in terms of cardiology, the other aspect that I mentioned, that how can we design basically better data sets so that we can enable AI tools. I think that is quite key, and I hope we will see more and more efforts in that direction in the future. At EPFL, together with the University Hospital of Lausanne, the University Hospital of Geneva, Unisante, we are trying to build a cohort, a longitudinal cohort of people, not necessarily patients, that would monitor for a horizon of 4 years in order to be able to see how the plaque will develop. There is a natural development of a plaque in each of us. So targeting a specific population and being able to get every information related to genomics, imaging, CT imaging, but also lifestyle data could be quite useful in that direction. So I would like to finish by acknowledging quite a lot of collaborators. I think what I presented today was quite quick, but it's a joint effort with many, many different people involved. The main work that I presented was the work from my PhD student Jeremy, and also Shao, who was a postdoc, but now he's a faculty in China. And also quite a lot of collaborations, SY Switzerland, Bernadette Sols, as I mentioned, she's our collaborator in the TDA aspects. In terms of health, we are collaborating with quite a few university hospitals here in Switzerland. And of course, I would like also to thank all the funding agencies that are involved in supporting these initiatives. These are some of the references of the different topics that I talked about. And thank you for your attention, and I'm happy to take any questions.
So yeah, thanks a lot, Dorina, for this very nice overview of different biomedical AI applications. So I think with this we can close the recorded part of this session, and then we can move to the questions.